Alright everyone, I want to talk about the absorption of carbohydrates. And I want to start here by talking about the sodium-potassium pump. Enterocytes, or small intestine simple columnar cells, which are the ones doing the absorption or involved in the absorption, they have sodium-potassium pumps on their basal membrane. And so it's pumping sodium out and potassium in, and this is going to create a sodium gradient within the cell. We're later going to use that gradient to get glucose into the cell, alright? And this is primary active transport, right? You remember that from chapter 3? Primary active transport. Later when we use it, it's going to be called secondary active transport. Because it's using energy, but it's using energy created by the primary active transport. It's using, the energy is the chemical gradient created by the primary active transport, which is down here, sodium, potassium. Alright, so carbohydrates, really the chain would be much longer than this. We have to break it down somehow. So how are we going to be breaking it down? Well, we have salivary amylase, but the food doesn't stay in our mouths long enough usually for that to really break down the carbohydrates. So, pancreatic amylase in the small intestine is going to be doing most of the breaking down, or the breaking down of the polysaccharides. So it breaks them down, but let's see over here. See, I have a dimer still. And what we need is, remember how these enterocytes, the small intestine simple columnar cells, they have the microvilli, and on the microvilli they have enzymes. So it's on their brush border. So brush border enzymes, I drew them in a maroon color. And these are going to break it down. So here I have glucose and fructose broken down to their monomers. And then, so once they're broken down to a monomer, we do that secondary active transport. Right, so they're broken down initially by pancreatic amylase. Then on the brush border, broken down into monomers. And once they're monomers, we can use that gradient, that chemical gradient that we established earlier, and pump a sodium out and a glucose in. Does that make sense? Because if you remember, is glucose hydrophobic or hydrophilic? It's hydrophilic, right? So carbohydrates, hydrophilic. So they can't get through that membrane like hydrophobic molecules could. So that's why we need this gradient, and we use the gradient, secondary active transport. Sodium out, glucose in. Now there is an exception. Fructose travels in by facilitated diffusion. Both of them cross by transcellular transport, going across the cell, out the basal membrane, and they can both diffuse into the blood. They're hydrophilic, water-loving. They can get into the blood. Does that make sense? So we have a few critical points, right? The sodium-potassium point, primary active transport, establishing the gradient. We have the glucose and sodium transporter using the gradient, secondary active transport there. Fructose is facilitated diffusion, and we have our brush border doing that final enzymatic reaction. It has, so I have a question. If sodium potassium pumps suddenly we didn't have any or they weren't working, would we be able to get glucose would we be able to absorb glucose? We would not, would we? So it's very fun that this is kind of starting to combine many different things that we've learned throughout the whole year. Alright, let me know if you have questions on carbohydrates.